Hello, my name is Sibu Mukwakwami. I am the Careers Associate at Education Matters. And as Education Matters, we are excited to bring you this podcast where we talk to different people who are working in Zimbabwe, interning in Zimbabwe, all professionals who've always been in Zimbabwe. But the whole idea around it is to help students and professionals who are planning to come back to Zimbabwe so that they have a soft landing when you come back home, because we would have prepared you through these conversations on working when you relocate back home in Zimbabwe. I'm so excited about the guest we have today, Rosemary Moyo, who was interning at AiBST. I remember going with Betty Mafemera for that employer meeting, talking to AiBST and trying to match her with this placement for internship, and they were really excited about having her, and they were talking about the past intern that they had also, and just how much great work the person had done. And yeah, I'm just excited to hear from Rosemary what was her actual experience. Rosemary, welcome. Tell us a little bit about yourself as well.
Thank you for having me today, Sibu Cecilia. I am Rosemary Moyo. I was an intern at AiBST. I just finished my internship. I am an undergrad student at the University of Connecticut, a rising junior getting into my third year. I am a biomedical engineering major specializing in computational systems biology and a minor in electronic systems with electrical engineering, focusing more on the devices and circuits part of electrical engineering, but as a minor degree, not a major degree. I'm also a founder of a nonprofit that focuses on mentoring STEM students, preparing them for sciences within Zimbabwe. I also do venture capital within healthcare, health tech, biotech, but within the States. So it has been a very nice journey also getting to see what Zimbabwe has in terms of the venture capital space.
Wow, that is so exciting. You know, Rosemary Moyo, I've heard people talk about how women should have like five channels of income. And when you were giving your introduction, I'm like, that's you. Look at you. You know, you have an NGO, you're an I want to say engineer, but I think that's like your minor. All right, I don't really, yeah. And then,
A biomedical engineer,
sorry,
a biomedical
a
engineer, most likely,
biomedical engineer with a minor in electronical engineering.
yes.
Wow. And look at you, you're into venture capital as well. So amazing, so inspiring. I'm really excited to have you on this call. And you know what? Feel free to talk about all these, your holistic. I want to hear more. Like, I know we have questions. We're going to talk about this, but don't limit yourself to my questions. Tell us about VC. Tell us about, your NGO as you talk. Feel free. I'm here for all the tangents. So, Rosemary, what inspired you to study biomedical engineering and why specialize in computational systems biology? Educate me.
Funny enough, I
Need to
never
speak a bit
wanted to.
louder so that people can really hear you. Try to match Sibu's loudness.
No, it's okay. I think it was where I had placed my microphone. But
Awesome.
funny enough, I never really wanted to be an engineer. I always would tell my parents that I was going to be a medical doctor,
Mhm.
particularly during cardiothoracic surgery, because I didn't want to be like my parents. My dad's a mechanical engineer, and my mom is an electronic engineer, electrical engineer. So I just had the stigma started, 2020 when I was in my fourth year of high school. I said, I don't want to be like my parents. I want to open my own path. Fast forward after A level, 7 months of working at a clinic because I got some few health certifications here. I was like, no, I I just want to use my certificates. Then I started working at a clinic voluntarily for about 7 months. Within those 7 months, I attended to more than 1,000 people,
Wow.
30 or 40 something people per day, doing drug dispensary, doing order sheets for medicines, also taking care of people who are sick, doing HIV and AIDS tests, diabetes, high blood pressure tests as well. So I figured within those 7 months that it was wholesome when I got to meet those patients outside of my work, or maybe when I'm in town on a weekend that they say, oh, Ness, in Zimbabwe, they say, Ness, the medical medications you gave me, they worked. Don't be honest, you took care of me.
Oh,
Those were like the
wow.
heartwarming moments. But then when I look back at the work, some days I'd get to knockoff time, which is 5 p.m., and I'd just feel sirens in my head. I just felt I can't do this anymore. Then when you see people, feeling happy, being well and better compared to the first time when they came for assistance from you, you feel that you feel that touch. But then I also realize, as much as I want to give, because I was only 19 at that age, and I was, when you're doing a job, it doesn't matter what age you are. That's the job you're doing, and you have to commit to it. So everyone was treating me like a nurse, of course. I had to give so much of me, and then I realized, yes, this is a job that has me giving back to people, which is something that I love to do. But I'm a person who's motivated by innovation. Stuff has to be moving around. I am not quite happy when it's the same things every day. I like getting excited. I like my mind being stimulated to think in a different light. And within those 7 months, I discovered biomedical engineering. I said, I could branch medicine and engineering and do innovative stuff that's fun, but also applying that to the healthcare space. And if you look at our country as well, we had a point where we only had one radiology machine within the hospitals. And that's not because we didn't have other radiology machines otherwise, but we have so much equipment in hospitals that needs biomedical engineers to fix them, or needs biomedical engineers to make new devices that work with our country, that work with what we have. So I said, biomedical engineering isn't bad. I can do biomedical engineering, and then I can see how I can apply that to the country. Then I was a bit torn between doing systems imaging and instrumentation for BME or computational systems biology. But then I realized I needed the software part, which I can call the brains. I needed the software part, the computational part to model biological systems. Then on the other hand, I also needed the body, which is the electronics part, the control systems part. So I ended up saying, no, let me do a major in BME and let that be computational systems biology, which can be applied to diagnostics, the pharmaceutical space, imaging as well within the medical devices, or organ and chips, the small portable labs. You can do that as well with the computational part. Then I could do a minor in electrical engineering, which gives me the body, which is more of the the device, the circuits, how the device works, so that when I go into a field, I'm not just going with a computational mindset, but I also understand the hardware that I'm using. And if I need to make any innovation, I can easily branch both the computational and the hardware part to work together to cook something up, bring something nice.
Wow, Mbuyanes, you got the brain, you got the body. Oh my goodness. Well, so wholesome, so holistic. I am so inspired, so inspired. Such a privilege to talk to you. So tell me, how does your minor in electronics and systems enhance your biomedical work? And I feel like you've already kind of talked about it, telling me about
Yeah.
the brain and the body. But yeah, maybe a little.
So most of the times when people think about biomedical engineering, they think about medical devices. But if you go deep into biomedical engineering, you figure out there's medical devices, which is the biomechanics or systems imaging and instrumentation. You also have diagnostics, which is the computational part. And you also have tissue engineering, prosthetics. That's another subfield within biomedical engineering. So instead of having my biomedical engineering degree be mainly focused on the medical devices, I wanted both the device part and the computational part. And my options might have been to do a double degree, but then honestly, like I might not really need a double degree. I just need a part where I can focus on the devices part, and a part where I can focus on the computational part, and having electrical engineering as a minor. Because, when you get into the job market, when you go into R&D, like research and development for, be it imaging or be it systems, you're going to be working with both the software and the hardware. It's not just the software, unless if you go into fields that have to do with diagnostics and you're just focusing on the computational part, yes. But if you go into R&D, research and development, you have to be well-rounded. You have to be an asset. You should be able to give something. And within biomedical engineering, if you have the knowledge about devices coupled with computers, it's not hard. It won't be hard on you. You'll get to, smoothly move into different roles and feel comfortable, and also understand when you are working with people from within different sections of biomedical engineering. So when it comes to my minor and the electrical engineering minor, it's heavy on the devices part, which also supplements my software part. Because when you start making devices, you start realizing, yes, you make a device, but if you're making a device for healthcare, you need to know how to code. You also need to know circuits. You need to know the circuits, and you also need to know the software part, because it works hand in hand. Imagine those Da Vinci surgical devices that now do surgeries for surgeons, while surgeons just control them on a console. That's electrical engineering plus the computational part joined together, because the devices parts, the control systems, it's going to be the machine itself, and then the computational part is now the brain of the machine. So my minor gives me the body, as I had once indicated, and then my major gives me the brain.
Wow, très cool. As they say in French, that is so cool. Summarize a little bit your internship, and like, what was your experience on a typical day in the lab at AiBST?
A typical day in the lab, it honestly depended on the stage. It depended heavily on the stage where I was with my research. But the first few days, it was heavily on making extractions from different solvents based on volatility and the types of bioactive compounds I wanted to get, grinding, drying, that's mechanical stuff, and drying too. And then after drying, it also moved on to reviving the microbes. In this case, I was working with mycobacteria and fungi, so you had to revive them at different temperatures. And then when you revive from the glycerol stalks, which are like small stalks that are frozen, that are stored within Eppendorf tubes, you then have to streak on agar plates, those plates where they show different colonies and then they say, oh, the colonies grew on agar. Yes, you then have to streak on agar to make sure that whatever you revived is the actual microbe that you're interested in, and it doesn't have any contamination. So if there was a contamination, because the agar plates needed 48 hours, revival needed 24. So if something goes wrong, you'd have to go back again and fix that. Then after that streaking part, you then move on to broth microdilution, where you're now looking for the minimum inhibition concentration. This is where those, high school pipetting skills came in handy, where your hand just had to be accurate within, because you'd be plating in small plates that have 96 wells, trying to put 100 microliters of whatever you're putting in those wells, and making sure that you put into the right wells, you leave clean wells clean, so that you can get your minimum inhibition concentration, because that's where you'd test if your extracts, like my extracts that I was working with, if the C80 or the E. absinica extracts were effective against the microbes that I was testing them on. So the broth microdilution at times could take a week or two, because if you have contamination, you just have to restart, because you want to get accurate results before moving on to the computational part. And after the broth microdilution, that's when you then go to the computational analysis. That took about a week. That was just making some graphs in GraphPad Prism to see how your extracts were behaving with those microbes within these small wells. But you'd use absorbances. For that, you'd use a spectrophotometer when you're incubating them to measure the absorbances. Then after all has been done, that's when you can make those lovely graphs. I have a research paper that is to be published, I think, within August. We're trying to do some peer review on it. So when it's published, I can share the link and people can, view, or people can just check my LinkedIn, my LinkedIn posts. I'll probably put a link for that journal there so that they can also see the graphs of what I was talking about. But that was mainly the lab. It sounds simple, but it was really hectic.
Girl, like I was going to comment to say, I think you would make a great professor because you have the ability to like say in simple terms things that I know are very complex. Like I can totally tell they're complex, but you say it in a very simple way that a novice like me can really understand. So that is so cool.
I was once a nurse, remember?
Yeah, Buyanis, you're good at it. So tell me something. So this paper that you're going to publish is in partnership with AiBST.
Yes.
Wow. So not only an internship, but a whole publication, girl.
Yes, because my topic was a combination of two master's topics, but then now doing vice, what we usually say in Shona, vice versa. Now taking, because one topic, the initial one was testing the effects of E. absinica on fungi, which is senior formans. Then the other one was testing C. zeri extracts on Mycobacterium M. smegmatis. And for my project, it was now testing E. absinica on mycobacteria and C. zeri on fungi. So instead of testing E. absinica for antifungal bioactive compounds, I was now testing it for antimicrobial active bioactive compounds. Then for C. zeri, it was now testing it for antifungal bioactive compounds and also checking to see if both plant extracts have dual activity. Can they both be antifungal and antimicrobacterial?
Wow. Talk about research that is like real life impact, right? I am just excited about this. Wow, wow, wow. So tell me something. Like I really want to know what is like the dual action of plant extracts on fungi and mycobacteria? Like why is that important?
So from what I just highlighted,
When I think
testing the
yeah, yeah.
yes, the plant extracts, testing them for dual activity. In this case, I was trying to figure out if the plant extracts had both antifungal and antimicrobacterial effects. And I was using M. smegmatis and Cryptococcus neoformans as my microbes in these experiments. And the reason why we wanted, you'd want, not we, but you'd want to find, maybe a bioactive compound or a drug or possibly a hit or, something that alludes to a hit that has a dual capacity to do both antifungal and antimicrobial. That means if you're to develop a topical cream or a drug, you're only going to need one thing. And for example, you would probably need a bioactive compound from E. absinica. Then you'd know that bioactive compound can work against C. neoformans and can work against M. smegmatis. Or C. zeri can work against C. neoformans, which is fungi, and mycobacteria as well. That means if any of the bioactive compounds within these two plant extracts can do both functions, that means when you're now developing a drug or a tropical cream, you're going to save costs as well when you're developing the drug or when you're developing the tropical cream. That is part of the dual activity. I'm sure you can relate to medicines that, when I drink this pill, it's going to cure my flu, it's going to cure my maybe hypothetically, let's say tummy ache, my running tummy, as well as flu. right? It's almost close to Tylenol, a pill used in that you'd find when you have flu. Maybe you have a runny nose and you have a sore throat. Then they give you some Tylenol before drugging you with heavy drugs. Then you just say, "Oh, I just drank Tylenol. It reduced my fever, and my sore throat is gone." You see, it's doing two things at once. You're still on mute.
Thank you so much. I was so amazed by what you were saying. I stayed on mute. I was gonna say, I think we can take one more question before we go on break. But I wanted
Yes.
to ask, like, so, what were some of your most exciting findings, and how did you combine, the wet lab and computational work? Yeah. And I feel like some of these things you've touched already. If you feel like you've touched it enough, we can move to the next one.
Yes, I'd say my most exciting finding was finding that DCM extracts for both plant extracts were very effective. In this case, I was testing Caesieri on fungi and Eabsinica on mycobacteria. And on both plant extracts, my DCM plant extracts had the most effect against both fungi and mycobacteria, which could allude to, just getting an idea of when you now want to develop a drug or a topical cream, you're most likely going to want to do DCM extracts to get your bioactive compounds. That was very neat and very fun for me. I did enjoy that, seeing the graphs and, of course, checking my pipetting skills, which weren't too perfect, but were fine. And then finally taking that to the computational part, because I think that we had a task to solve a problem within the organizations that we were working on. I decided to work on helping the master students with their LCMS process, which is a process where you now take those bioactive compounds, the extracts, and then you run them just to see which bioactive compounds are within those extracts. So initially, when I got the LCMS data, I realized the students were going row by row, checking to see if the bioactive compound had antifungal or antimicrobial or both effects. But then these are Excel sheets with about 3,000 and something bioactive compounds. I surely wasn't going to do that. Even if they were 100, I was not going to go row by row in Excel. So for the challenge I solved for that, I'm currently developing a manual with some Python stuff that they can use using RDKit, which is a cheminformatics big library that has lots of stuff to do with chemistry, bioactive compounds, smiles, all that in there that they can use, which I used for my computational part to screen those Excel sheets with 3,000 and something compounds and just get the top 30 compounds with the most similarity that you want for antifungal, antimicrobial, or dual activity. And then it just pastes it to an Excel sheet that's stored on your computer instead of going cell for cell and editing it in there. So that was the first thing I did when I took the data from the bioactive compounds. I took mainly the compounds that had the most effects, the most effective compounds. And then I ran them within my Python code using RDKit. And I had my top 30. From those top 30, I just took top five because usually you'd want to score that's 85%. But in this case, I didn't get 85%. Most of my percentages were 20 something. Then if I got a 30%, I was like, okay, this is so good. But it also means there's potential for further research within those areas as well. That was the first thing I did. Then after taking the compounds that had the most dual similarity or antifungal similarity or antimicrobial similarity, I then took the most similar compound. Then I went online, went on Swiss Target to find some targets within the compound, within the human, because Swiss Target has a range of proteins, genomes and stuff. So when you put the compound in there to find targets, you are trying to find what that compound mainly targets within different species, be it humans, be it bacteria, be it there's a wide variety. And then I would take note of what it affects the most. If it's proteins, then I would check, because if it's proteins, it also would show you which protein is most likely going to be affected by this compound, which means most likely that's the protein that's going to be disrupted by this plant compound. Then you take note of that protein, you'd go online, you'd then go to like UniProt so that you could get a sequence of that protein. And after you get the sequence, you change it into a protein database file. You'd change it into is it PDP or PBD, so that you can now model it. You can see it in Python stuff, see how the molecule is, then prepare it for docking. Then if you're preparing the protein for docking, you're now trying to see, isn't it online Swiss target said this compound is most likely going to affect this protein within this organism. Now you want to see what exactly does this compound attach to, and how does it attach to this? What type of bonds does this plant compound attach to within this protein? So that you can see how this plant works in disrupting whatever it's disrupting within the compound. So I finished that. I did see some scores, some hydrogen bonds, lots of hydrogen bonds, and lots of nice pictures as well, scientific pictures. Those ones you used to see on textbooks. Yeah. So that was mainly the computational part. It is complex. I can't really explain the whole process, but I think and I hope that gives people an idea of what the computational side looks like.
Wow, that is so cool. So cool. Um, I think we'll take the next few questions in the next after the break. But I
Yeah.
would say it's so cool. Mbuyanis, you have the brain, you have the body, you got a publication, you're working on a manual. That is so cool. Who gets all of that within like one internship period? How many weeks was your internship?
6.
Girl, this is amazing. Amazing. All right, let's take a short break and then we will talk more about the impact of this type of work, even for the Africa region and all of that. So, yeah, for now, just a short break. Hey, Rosemary, I'm so excited that we're back from our break and I'm going to hear more from Buyanis, the one with the brain and the body. Someone who I really feel is holistic, who probably has more than 5 channels of income, who's still studying, who was just interning and during her internship managed to do a publication and is actually working on a manual. How amazing. Talk about impact. Okay, Rosemary, tell me something. If your research becomes a drug or a cream, how could it impact healthcare in Africa?
So if we look at the organisms, the microbes that I was researching on, we find that Cryptococcus neoformans is the cause for about 180,000 deaths, mostly from sub-Saharan Africa. And M. smegmatis is mainly in the skin infections area, but is rarely recorded. But whenever it's recorded, it's fatal. Then for C. neoformans, it's mostly cryptococcal meningitis and cryptococcal pneumonia, which could come as pneumonia, but it's actually caused by cryptococcus, or meningitis, which is caused by cryptococcus. Most patients being from either the meningitis or the pneumonia. So if this research was going to be, or is going to be furthered to give us maybe a topical cream for M. smegmatis and maybe a drug for Cryptococcus for the pneumonia and meningitis, it would have a big impact when it comes to reducing the number of wait a minute. I think my camera just clipped. Can you see me?
I can see you and it's fine right now. Let's go.
Oh, wait, okay. So back to what I was saying. I can start afresh. So for Cryptococcus neoformans, it causes about 180,000 deaths recorded per year from mostly from sub-Saharan Africa through meningitis or pneumonia. Then M-smegmatis rarely recorded, but whenever it's recorded, very fatal and results in about very bad skin infections for its patients. So if we're to get a topical cream for M. stigmatis, that would really alleviate the patients that have to go through that excruciating pain. And for senior formans, there could be development of a drug which could work against senior formans in the lungs, against pneumonia, and cryptococcus neoformans, cryptococcal meningitis as well, which can do both jobs at once. And if that's going to be developed, I would say it would really be a good health solution as well as a hit within the African market, particularly sub-Saharan Africa, reducing the number of casualties we have to Cryptococcus neoformans. Because as you know, bacteria and fungi, they're also living organisms. They go through evolution. They grow resistant to stuff that inhibits their growth as well. Similar to the human immune system, which fights against diseases and upgrades that the bacteria and the fungi, they also upgrade to develop mechanisms to guard themselves against the antibiotics that we already have in the market. So this is mainly coming from the traditional approach, now getting into science.
Wow, that is exciting. So, so exciting. I've always had like skin disease growing up. I mean, it's acne, but like since I was 12. And at some point I wanted to be a dermatologist just because of, you know, what my experience has been, what it has done to my
Yeah.
self-esteem at certain points, and even like the bullying I've experienced because of that. So yeah, it's amazing to realize that some of these things, it seems like, oh, it's just biomedical engineering. Oh, it's only for health. But it really even affects people's lived experiences. Like I'm saying for myself, like, I've experienced bullying, I've had low self-esteem from these things. So it's amazing. The work that you're doing has greater impact than we want to realize at certain times. Um, and I'm thinking, so how can traditional medicine and plant research contribute to modern healthcare innovation? You know what, Rosemary? You remember when COVID happened and
Yeah.
what were we using in Zimbabwe? I mean,
Zumbani.
you were in the States. What were we using in Zimbabwe? I was in Zimbabwe.
I was also in Zim. At that time, I was in my fourth year of high school. It was Zumbani. Most people using
So
Zim.
many, all of these things. Yeah, yeah. So that's kind of my question. Like, yeah, how can traditional medicine and plant research contribute to modern health innovation? Yeah.
So if we look at the project that I did at my internship, we didn't just pop out with those plants to just say, oh, there's E. absinica, let's test it. Or there's Caesieri, let's test it. Within traditional medicine, people had already found out that the leaves of Caesieri had medicinal effects. So people can grind them, make tea with them, same as what people were doing with Zumbani leaves. And then within traditional medicine as well, the other plant, Aeopsynica, people had found out that they could actually use the bark to treat wounds, etc., right, within traditional medicine. And if you do research online, there's lots of work covering Absinica and Caesieri used within the traditional space. So now it was taking those plants that are used traditionally to now say, instead of us just saying, if you grind the bark and if you put ABCD, it works. let's now scientifically validate this. Let's now scientifically see what's in these plants, what bioactive compounds are in these plants that have these effects. Can we harness these bioactive compounds to mass produce something that can easily be used, but taking from traditional medicine to now you'd be getting into the field of phytomedicine, which is for plants, right? But not discarding the traditional medicine information that we already have, but just adding on the work that has already been done, figuring out that this plant actually has some effects. And then I would say, I personally feel when it comes to the field of drug discovery, I feel traditional medicine works hand in hand with the scientific approach. They could benefit from each other. The problem is when most people think about drug discovery, they just think, oh, we're just going to use science. We're just going to go into the bush, find something that we think works and experiment on it. But you have, I think there's this board here in Zimbabwe. I've forgotten the exact abbreviation, this board of traditional healers, where they have a directory of plants that have been used for decades by our ancestors and are continuously being used, new ones being added till this day. Why would you just go into the bush and start hunting for things? Go to the people who are using traditional medicine, ask for their databases. You might find something that's very intriguing, or something that can give you something more to a problem that we might have already within the healthcare system. I'm sure after COVID, now you see more people are welcoming to traditional health medicines when it comes to tea leaves that were never branded as tea leaves before, that were thought of as weeds. People are now more welcoming to say, oh, I can have herbal tea. We can use the herbs to put on my wound. But before COVID, some people would not want to be associated by using herbal tea and only wanted the scientifically developed drugs. But you see, it's a field where they can work hand in hand to produce something bigger, which would also be beneficial for instance, countries like our country, Zimbabwe.
So awesome. I really like the idea of validation. And I think one of the struggles that I've heard, because I love teas, I love herbal teas, I love healthy stuff. But one of the problems is dosage, right? Like, okay, you go and take Zimbani, but like how much of Zimbani? So, this
Yeah,
scientific approach is so important. It's so, so important. I'm excited about the work and the research that you're doing. I would like to know, like, what did this internship teach you about Zimbabwe's potential in health tech and pharma? Yeah.
I would say when it comes to this internship, with my experience, it wasn't much on showing me the potential of Zimbabwe's healthcare, health tech, and pharma, because the potential was already there. Wherever you have a problem as an entrepreneur, there is an opportunity to make something there. You see, we have a problem of medical machines. There's an opportunity to be an entrepreneur focusing on medical devices, and you can flourish there. I worked as a nurse for about 7 months, and within those 7 months, we'd have a shortage of drug supply. We would have a shortage of simple drugs such as paracetamol, ibuprofen, the simple painkillers. That means if you have simple painkillers being in shortage, you can't expect the complex drugs to be in surplus. That doesn't work, and that's another field of potential. Having our own pharmacists, having our own drug discovery, drug production facilities, right? These are different places where we could get the potential there. But at this internship, I would say what I saw was the amount of private stakeholders that we have that are open to committing to development within the country, particularly development in research, because that's a field that I wasn't pre-exposed to. I wasn't so sure about there being investors that are really interested, or individuals that are working within research in Zimbabwe who are interested in expanding the field of research within Zimbabwe, be it for public health or for stuff such as genomics, therapeutics, which is something that really opened my eyes here, because I didn't know about AiBST. I got to know about it through the internship, and to also know about the other health research programs that are within the country and within the region that have so much potential to do great work and solve problems that are for Africa. Because, some people get heated and touchy that, oh, those developed countries are not developing drugs for countries such as ours that are in developing countries. No, they have their own problems as well. They have people who have heart challenges, stroke. They're trying to solve problems within their own communities as well, such that when it comes to our communities, they're going to try and help our communities after they've tried to help their own communities. So seeing the amount of research that's happening in the background, in the private spaces, the amount of scientists that we have that are just doing background work, I was impressed and I was overjoyed. I mean, I don't think they even saw the amount of joy that I had seeing them do that work, seeing them do the impacts that they're doing in solving issues such as cholera or malaria. In some countries, if you speak of cholera, people get shocked to say cholera in this day and age. But we have cholera affecting us now, and we need our own scientists to be doing the work to help our own community. So I would say that's what I really saw where I was working, and that made me happy and inspired too.
Wow, amazing, amazing. I would love to know like what technical and like soft skills helped you to succeed in this internship, and how did your electronics knowledge support your work? I think you touched the other part of the question. Maybe you can focus on the soft skills and technical skills that you used while on this internship.
Yes, so for the soft skills, communication, of course. Nothing goes well without communication. Even if you're a scientist, you still have to communicate. If you're an engineer, you have to communicate. So I would say heavily on communication, being a team player, being able to work with the people in teams. When it comes to technical skills, it was heavily coding. It was heavy on coding, having Python knowledge, because if you look at my computational part, which you'll see when I publish the paper, almost everything in my computational part was Python. From what I used to screen the compounds, I used Python. RD kit, Python. When I moved from screening compounds to now having the structures in PyMol, which is Python, Pi for Python, and then molecules, we're using Python for visualizing molecules and cleaning them. That was Python. From now taking the visual part, going into Autodoc, docking, lots of it, the backbone was really Python. But I would say I had to really, besides understanding Python, some skills that had to be useful were, because of course, yes, you can have the Python, but you also have to have RD kit skills. You have to know how to use RD kit. You have to know how to use Autodoc. You have to know how to use Pymol, which really isn't difficult. I mean, you can, go about it in different ways. You should know how to code, how to use Anaconda, which is like the suite that has different apps in there. You have to know how to use a Jupyter notebook. So it was a lot of that, but the backbone being Python, because I already had experience with coding. So now using stuff within the space that had Python as a base, that really, really helped me a lot.
Wow, so cool. So cool. I would love to know actually, like, why did you choose to intern in Zimbabwe? I mean, you could have interned in the States or other places. And how was it different from labs abroad?
I decided to intern in Zimbabwe because I needed to have an idea of what the Zimbabwean market looks like. What does the space look like? What's missing in the space? what's there in the space, almost similar to what I like doing with venture capital. As much as I do venture capital in the States, I also got interested in what the VC space is like in Zimbabwe, which I got to learn from connections as well, talking to people, having interviews. Interning in the States, that's a given. Third year, your summer for third year, you will intern in the States. I didn't need to rush that. I needed as much international experience as I could get, solving problems that are in my community, problems that I can easily relate to before doing stuff that's heavily for the States. Because something that I also really got to appreciate is that even if you intern away in a different country, the skills that you gain in that country, you're going to use them when you go back to the States to whatever internship you choose. You're going to use them. But if you can find a space where you can work and do a project where the work is real life affects your communities, you can easily relate to the challenges, it's easier. You also enjoy the work, besides also getting a holiday back home for the food and family. If you grew up I grew up in Zimbabwe. I can easily relate to problems in Zimbabwe when people say, oh, we have problems with cholera, we have problems with malaria, we have problems with meningitis, pneumonia being caused by this mycobacteria. I can understand. And if I'm now using the skills that I get from school here, I can now see how useful those skills can be. Because when I was coding Python in classes, it wasn't Python, the type of Python I used for my computational part. Honestly, if I have to be honest, all the tools that I used within my research, I have not used any of them in school.
Wow.
I've only used them during my free time out of curiosity.
Wow. 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 Wow.
Out of
Always
curiosity, but not
hard.
that I had that in school. In school, I was just taught the basics of coding.
Wow, I like that. Like, when I have one-on-one sessions with students, or young professionals who want support for their CVs, I tell them a lot that, it's not about editing the CV so that it looks good. It's about doing the hard work and then just putting the hard work you're doing onto the CV. Like
Yeah.
going extra mile. It's not enough
Yes.
what you're learning in the classroom. Go, out of curiosity, explore further. And that's really what makes you like, different and yeah, and
You have to
memorable.
explore
Yeah.
because I did not think I'd be screening thousands of compounds. As soon as I saw those Excel sheets, I said my computational part is going to work today. And my supervisor was laughing at me. He was saying, Rose, don't you think your wet lab was already a lot? And I said, no, I did the wet lab, but let me do the computational part. Let me screen these things. Let me autodoc them so that I see how they interact.
Wow.
Because I am not going to be going row by row, looking, copy pasting into Google. Why don't I just use RD Kit, this big library, but that has chemistry stuff in it that's used for chem informatics? Why don't I use that? Why don't I use the smiles? Because in chemistry you have molecular structure, and now we're just using smiles, which looks like is it a formula, but then it's not really, because it shows how the compound looks in 3D. So if you have a computer, it can now read to say, okay, this is the smile. I can translate it. This is what it's supposed to look 3D.
Wow. So tell me something, Rosemary, what do you see as like biomedical, yeah, like maybe both opportunities and challenges in the biomedical field or innovation in Zimbabwe?
I would say when it comes to challenges within the biomedical field, the first is knowledge. Knowledge of the space, knowledge of what the space has to give, besides equipment, of course, besides equipment, and also being open-minded about careers, because when people think about engineering, you're most likely going to think about electrical engineering, mechanical engineering. Yes, you can have a mechanical engineering and electrical engineering and apply that in the medical space. But you can go to school to focus heavily on just the because the medical space is not small. The field of medicine is too large that if you have a student that knows that they're being trained to develop devices for this field, within those fields, the student will probably find a subfield. That's the easier things can get. I also think having companies that actually do hire biomedical engineers, because it's fairly a new field here, we would need some companies. There's already one company that's there for biomedical engineering engineers for devices. I heard of another one which is mainly the instrumentation part and systems too. But we would need more companies that would like to hire biomedical engineers, besides support and initiatives that push students to be innovative and students to feel comfortable being an engineer for medicine.
Wow. So cool. So I'm just wondering, like, what advice would you give to Zimbabwean students who are abroad and they're considering to return to Zimbabwe for internships? What would you say to them?
I would say do your research. Research, explore. You don't have to explore in person. You can explore online, look for people who are doing the work in those fields, do some research on how the landscape looks like for Zimbabwe, so that you know when you're coming back to Zimbabwe what type of area would you want to explore. 9 times out of 10, you'd be shocked when you get into that field and discover other new things that you didn't know existed. But do your research, talk to people, ask for phone calls, ask for meetings, also check people's LinkedIn's as well. When people post the impact that they're doing, that's another place where you can find out stuff that you can really use. Talk to people who've interned in Zim, talk to people who've studied abroad and have returned to Zim to have a career. Like the founder of AiBST was a student in, was it Ireland and then the States, worked for AstraZeneca, one of the biggest pharmaceutical companies in the world at the moment, and still back then. Now he has AiBST that he founded in 2002. It's nice to talk to people, individuals like that, because you get to know more about why they decided to come back, how they saw an opportunity, how they grabbed that opportunity and ran with it to the point where they now have solid stuff years later.
I love that. And I'm just thinking for yourself, Rosemary, like what's next for you and what like biomedical solutions would you love to see developed in Zimbabwe?
So when you say what's next for me,
Well, I know you're a student, but like I guess coming from the internship, like what are you bringing back into the classroom? Or you could answer it the way that you want also.
yes, I say that because I'm a woman of many colors. If I was going to be in the Bible, I would be, is it Joseph with a coat of many colors? Yes. So when it comes to what's next for me, of course, it's still running my nonprofit. It's still doing my venture capital stuff, but now I have a deep understanding of drug development, drug discovery, pharmacokinetics, pharmacodynamics. Not deep, but a basic understanding. So that would really help me with investments that we might look at. When it comes to skills that I'm bringing back with me, be it in the lab or in my education and my career, I'm definitely bringing back lots of computational skills with different softwares, Uniprot, Autodoc, PyMol, Swiss Target, lots of coding as well. Not just coding, but chemi informatics type of coding. I'm also bringing back lots of lab experience, broad microdilution, work with the spectrophotometer, dilution, lots of that lab work, as well as knowledge on solvents. Because when I started this internship, when we looked at the extracts, I did some literature review, and I was chatting with my supervisor, and I was giving him some reasoning as to why we used specific solvents to understand if my understanding was correct. And we're talking about volatility, and we're talking about polarity. So that's definitely some experience that I'm bringing in, because now I know when we're taking bioactive compounds using different solvents, we want to separate them based on polarity and most likely volatility. And just by knowing that, what am I going, you can actually use your intuition to say, from this basic knowledge that I know from basic literature, my results are most likely going to come out this way, in this order. So I would say that's more of the skills that I brought. Lots of communication skills as well, lots of write-up skills, because I had to write a publication that's being peer reviewed as well. So I would say yes, that's more of the skills that I brought, besides data analysis as well.
And you know, something that you're definitely bringing back that you're not saying explicitly, but that's coming like in all of your responses is relationships. Like it really feels like you had great relationships with your colleagues, with your supervisors. It's really coming out. So you have stronger networks and awesome. Good for you. I am so happy for you. Anyways, as closing questions, I'm gonna ask you two questions as we close out. And I would like for you to describe your internship experience in one sentence. What would you say?
In one sentence, I'd say my internship experience was unexpected, but was so exciting because I got to learn different things daily, intriguing sometimes, getting to also know from the students that when your plates are contaminated, you can also tell which organism contaminated your plates based on the smell, because believe it or not, some microbes smell like soil, some smell like something sweet. So if you have the smell, a certain type of smell, this microbe potentially contaminated my plate. That was so fun because I never knew that you could actually use sense of smell to detect microbes.
Wow. Well, I love your work. You use all the senses like your eyes, your nose, I don't know about the mouth, but definitely your hands, tactile. I love
Not the mouth.
it. I'm sure not the mouth. You may
Yeah.
die.
I was a bit skeptical, but smelling as well. But from looking at, because the place has masters and PhD students only, so just learning. Of course, they have more experience than me. I'm in my third year of bachelor, so I say, okay, so it's safe for me to smell. They said yes. What does that smell like?
Wow, that is so cool. That is so cool. All right, final question. So what is one key lesson that you're taking away from this internship experience? And if you built a startup in Zimbabwe, what would it focus on? So it's like a double-edged question. What's one
Right.
key lesson you're taking away from this internship experience? And if you're gonna start a startup in Zimbabwe, what would you focus on?
My key takeaway from this experience is that complexity isn't always complexity to make things difficult. If it's complex, yes, it could be difficult, but there's so much flavor, so much ingredients in there that you can pick out and have an amazing experience. I mean, I see one of the master students was doing endophytic fungi instead of the plant extract that I was using. She was taking a leaf with fungi in there, fungi that have a relationship with the plant, where the plant helps the fungi survive and the fungi protects the plant. And she would culture them, remove the fungi from I mean, they wouldn't remove, but she just culture the leaf, and the endophytic fungi that are inside the leaf would just come out on their own. The project was pretty complex, 28 days of incubation and extraction and stuff. But it was so fun because I got to also learn some stuff that I didn't know, such as antagonistic activity, which has more to do with plating of agar. And when you think antagonistic activity, the first thing you think is protagonist, right? But it was complex, just not complex for the fact of being complex, but complex in a way that's interesting and gives you a different view to say, instead of taking the leaf or the bark, we could actually just take one leaf and get what we want from just that one leaf. So I would say my key takeaway, as I had indicated, is that complexity is not always just complexity and difficulty. Complexity can be opportunity.
Wow. Complexity is not always complexity or difficulty. Complexity can be opportunity. I'll take that. I love your tagline.
Then
Wow,
when
wow.
the startups that I would start, I already have a nonprofit for we're trying to incubate scientists and science entrepreneurs there. I would say for a startup, I could start a startup that focuses on diagnostics. Diagnostics using medical devices and the computational knowledge, so that we wouldn't have lots of cues before you proceed to look for something. You know already the root, maybe a root cause, maybe not exactly, but just maybe, right? And maybe develop more devices that people can do at home testing that are cheaper, so that someone will only have to go for an extremely expensive appointment when they have to go for an extremely expensive appointment.
Wow. Rosemary, it's been really, really awesome talking to you. You know, on some podcasts, like I talk a lot, but today I was like slightly quieter because I was learning a lot. I really, really love how you explain concepts. It's so simple. Like, you make complex stuff easy to understand, easy to digest. I think you may consider being a professor, maybe in your 60s, 50s, 60s. Sixth channel of
I
income.
like 60s.
Yeah,
Well,
but
thank you for having me, Spusi Siwe. It was lovely answering the questions, and I hope the listeners will get to watch the podcast, the interview, will find some inspiration. They can feel free to reach out to me through LinkedIn. They'll just look for Rosemary Moyo. They will find me. I'll
Rosemary
be
Moyo, LinkedIn. Yeah,
yeah.
yeah, yeah.
So,
Awesome.
or probably they may find biomedical engineering, whatnot. But I mean, they would have seen my face, so they should know me.
Yeah, yeah, yeah. True, true. That is so awesome. Wow, it's been a mouthful. It was so beautiful having Rosemary Moyo with us as our guest on the Education Matters Zimbabwe Career Connect podcast. It was amazing. I'm sure you just learned a lot of stuff, simplified but really complex and cool stuff. I think even I will want my daughter to listen to something like that just for inspiration when she grows up a little bit more. But this was really for us to learn that when you come back to Zimbabwe and intern, it can really be something that is new and innovative and really, really interesting. And you can get a lot out of your internship experiences. Look at Rosemary, a publication, a manual, so much networking, so much knowledge on what's there in the market in Zimbabwe because she's also into venture capital. It's really, really amazing. I mean, on top of that, the cherry, right? The cherry on the top is she was also home with her family, just enjoying being back in Zimbabwe, eating sadza. Yeah, so awesome. Um, so this one is just to inspire anyone who's studying abroad, wants to come back home to Zimbabwe for an internship. Zimbabwe Career Connect actually has a well-organized internship program. Betty Mafemera is doing a great work with this internship program. And you have an experience like Rosemary had, an experience that you take those skills, those experiences back into the classroom, really makes you a better person for your profession, even in making other choices for your career. So I highly encourage you to apply for the internship experience when it comes out. If you're someone who's already a professional in Zimbabwe or in Africa, and as an organization you are willing to take internships up, please look at Rosemary Moyo. These are the type of people, the type of students who will bring to you capable of computational work, lots of different stuff. So, this is really, really a great example of what an intern would look like. We would love to hear from you if you're an organization, if you're HR or CEO of a company, and you want to take on board interns from Education Matters. We would really, really love to partner with you as well. So as we close out, big shout out to Rosemary Moyo. Thank you so much. Thank you not just for this podcast, but for the work you did at AiBST and the inspiration that it is to everyone who's listening and the type of doors it continues to open up for future interns.
Thank you
so much. All right. And then bye-bye for now. Until next time.